hyperplasia of the paraganglia, which is a precursor of paraganglioma. We did real-time analysis for glycolytic enzymes and mitochondrial genes in these knockout mice. And then in vitro we do siRNA-based knockdown of SDHP in cell lines, HeLa and A2058. A2058 is a neural crest tumor cell line, which is similar to pheochromocytoma in origin. And then we test on these cell lines for the lactate secretion, the lactate dehydrogenase enzyme, and for the bioenergetics using oxygen consumption assays and ATP assays. From the first results, in humans we found out that SDH mutant pheochromocytomas have a high expression of profile of the glycolytic enzymes and reduce mitochondrial enzymes and enzymes that are involved in oxidative phosphorylation, which indeed is Warburg effect, that is high glycolysis and reduced oxidative phosphorylation. From the set of the knockout mice, SDHP knockout mice, the mice which develop neural-derived tissue, we took them and did a real-time expression analysis, which showed that there was upregulation of the genes involved in the glycolytic pathway, like the glucose transporter, the triose phosphate isomerase, phosphoglycokinase, and lactate dehydrogenase A, and decreased expression of regulators of mitochondrial function like PGC1 alpha and SDHT was observed from the early postnatal ages through the adulthood. This is just a slide showing the efficiency of a transfection or knockdown. So the first we tested the efficiency of a knockdown using Western blot, and then the mitochondria were extracted from the SDHP knockdown cells and tested for the SDH activity, which confirmed that SDH, the sustained dehydrogenase activity, was less in case of the SDHP knockdown cells when compared to that of the control. And then we tested for the glycolytic activity of SDHP knockdown, which is increased lactate and increase in lactate dehydrogenase gene. The cells that were transfected with SDHP knockdown showed to have higher levels of lactate and higher levels of lactate dehydrogenase gene was also observed by real-time PCR in both HeLa and as well as A2058 SDHPI knockdown cells. The high level of lactate, it means that it's a high glycolytic activity, which is one of the characteristics of Warburg effect. Then we tested for the bioenergetics, that is oxidative phosphorylation, in the SDHP knockdown cells. The SDHP knockdown cells show that they were lesser sensitive to oligomycin, which is an ATP synthase complex 5 inhibitor, than when compared with the control, both in the case of ATP assays and the oxygen consumption, showing that SDHP knockdown cells are in fact less dependent on the oxidative phosphorylation for its ATP synthesis, which is indeed one of the characteristics of the Warburg effect, that is reduced oxidative phosphorylation. With all these three different study models, we conclude that all the three SDH disrupted models show a metabolic pattern of increased glycolysis and reduced oxidative phosphorylation, which is the hallmark of the Warburg effect. And sustained dehydrogenase loss is a faithful model of the Warburg effect and can prove critical insights for understanding the basis of this surveillance phenomenon in cancer. And finally, an altered metabolic profile in SDHP knockout mice suggests that this may be an early event associated with a single allele loss of sustained dehydrogenase. With this, I conclude my talk. I thank Dr. Satish Dalia for my supervisor for providing me this opportunity to coming over here and presenting my presentation, and I thank Dr. Finn for being an instant and visiting my colleagues at the Health Science Center of the University of Texas Finally, I thank the audience for patiently listening to my presentation.
Thank you very much for your interesting data. I was wondering, one of the first results you showed was on the human tissue.